Hello students, welcome to LEELA'S tutorial. So today we'll start with Plant Biotechnology. In Plant Biotechnology, we make use of tissue culture and genetic engineering techniques in order to produce genetically modified plants. These genetically modified plants consist of the desired gene of interest which we have introduced. Right? So like this you are going to produce a better variety or a new variety or an improved variety of plants. So let us go into the topic. Here the plant tissue culture is defined as growth and maintenance of plant cells, tissues or organs in vitro condition, that is under cultural conditions, under suitable environmental, physical, aseptic conditions. You are going to produce the plantlets using plant parts, not the seeds here, right? So here this was first performed by the scientist Gottlieb Haberland, who is considered as father of plant tissue culture. Right? He used Knopp's salt solution and he has inoculated the plant cells and proved that each and every plant cell is having an ability to develop into a complete plantlet, which is defined as totipotency. Right? Totipotency is the ability of the single cell to develop into a complete plantlet. Right? So from then the plant tissue culture has been used. Right? So here let us see what is the use of this plant tissue culture. The first one is in clonal propagation. If you have any superior variety plant, from that single plant you can produce a clone of plants. Right? So here in agriculture, if you are going to sow the seeds, all the seeds may not produce the same quantity of the product. Right? Whereas in case of clonal propagation, all the plantlets which are produced by using tissue culture will be having the same quantity. Okay, they will be producing the same harvesting capacity will be there. Then next one, it is the genetically modified plants are produced. So whatever the gene of interest you have, if you want to produce herbicide resistant plants, or it can be drought tolerant plant, disease resistant plant, or if you want a plant to produce the increased sized fruits, or you want to shorten the plant, the dwarf plants, or if you want to increase the length of the plant or length of the fruit or the vegetable. So all these can be done by modifying the gene and introducing into the plants, then go for the tissue culture so that you can produce genetically modified plants. Then another advantage is to produce secondary metabolites. So secondary metabolites, the products of plants like terpenes, or you can say the fragrance of the flowers, or you can say the medicinal oils from the leaves. So all these can be produced by using the cells. Right? Then next, production of haploid plants. Normally plants are diploid. The pollen and the ovum, the haploid gametes, undergo fertilization to produce a diploid embryo. Then that seed is used as a used in order to produce a diploid plantlet. But here we can produce haploid plantlets. Only n number of chromosomes will be present. That means you can make use of only the pollen. Right? Without fertilization, you can use the egg cells or the ovules, right, before fertilization, or you can make use of pollen or the anther culture, where you can produce single gamete plants. They are haploid plants. Then another advantage is production of somatic hybrids. Again, you are making of somatic cells. So normal leaf, from the leaf also, if you are going to take a plant cell, you are going to devoid the cell wall. So the cell without cell wall is referred as protoplast. So these protoplasts are fused in order to produce hybrids and hybrids. Then another advantage is to overcome the male sterility. So even the male sterility can be overcome by this tissue culture technique. Then you can increase the growth or increase the production or increase the length of the plants, and also plant tissue culture requires very less space. Okay, within small area you can produce thousands of plantlets which are having the desired gene of interest. So let us go into the procedure. Here it is the culturing of the explant. So in tissue culture, first main thing is selection of the explant. Explant is any part of the plant body. It can be leaf, flower, right, stem, meristem, root tip, shoot tip, right, axil, anything, nodes, internodes, any part of the plant which is used for the tissue culture technique is referred as explant. So let us take the leaf, right. So if you are selecting the leaf, you are going to perform surface sterilization process. Over the leaf, there may be some contaminants from the air, dust, right. So here all these contaminants must be sterilized, right. So here you are making use of mercuric chloride in order to surface sterilize the explant. Then you are going to rinse it in double distilled water, then air dry in laminar airflow chamber. Okay, then here you are making use of chemically defined synthetic media. So all the components are mixed in this media and then they are sterilized, or you can say they are sterilized in autoclave, that is steam sterilization, right. So once the media is cooled, you are going to inoculate the sterilized explant into this sterilized chemically defined synthetic media. So once you inoculate the explant, it is the incubation process. So in incubation is nothing but you are maintaining the cells in order to get adjusted to the new media and grow, right. So here you have to provide the controlled environmental or physical aseptic conditions, right, such as pH, temperature, light intensity, everything must be maintained which are suitable for this explant to grow. Then it will be plantlets will be produced. Okay, so not only by using the seeds but also in plant tissue culture, by using the explants you can produce the plantlets, right. So this is the procedure. If you see the diagrammatic representation, here you can see the first step, it is inoculation of the explant. Here the explant is inoculated in sterilized plant tissue culture media, right. The same thing, chemically defined synthetic media or plant tissue culture media, right. So it can be a solid media or a semi-solid media or liquid media. Here I have shown semi-solid media over which the explant is inoculated. So now it is covered because it must be maintained under aseptic conditions. So here now after inoculation, second step is maintenance or incubation. So all the conditions, aseptic conditions are maintained. So what happens here? At least one of the cell from the explant will express the totipotency effect. That means each and every cell or a single cell will have an ability to develop into a complete plant. So what happens here? Those cells will, the undifferentiated mass of cells are referred as callus, and these callus is nothing but a mass of cells. These are produced from the explant. So once this explant is forming a callus, an undifferentiated mass of cells, here I have represented as a red color but they are half white color, right, or cream color. So once the callus develops, again here you can see the green color dots which are nothing but meristemoids. So each meristemoid will further develop into a single plantlet. So here I have represented complete callus has been transformed into a complete meristemoid tissue, and each meristemoid will give rise to a single plantlet. Then you are going to transfer them into the rooting and the shooting media, where you get root system and the shoot system, where complete plantlets will be produced. Then you will be transferring them to the greenhouse, okay, where they get adjusted to the new environment, that is acclimatization. Okay, so this is the overall procedure of the tissue culture technique, right. If you have any doubts, put it in comments and subscribe for further videos.